Okay, so today I want to talk a little bit about developmental biology. Um, and this is the last lecture in the course. Good news to, I'm sure, many of you. Just one detail left after this, and we'll take care of that next Tuesday afternoon. But for this lecture, I want to focus on some of the uh, concepts in developmental biology. We really will talk mainly about very early development. Next spring in the oral histology class, you'll be talking about oral development, and I hope that some of these concepts will be familiar to you when you look at how oral tissues are um, developed in humans. So we'll be talking about a lot about using model systems for studying development, and you'll see it's amazing how similar uh, different organisms are to each other in terms of the signals used to drive the development. So let's look at some of the basic processes going on here, and there are four essential processes uh, to make a multicellular organism shown here. And of course, one is cell proliferation. If your cells don't divide, then of course they're not going to populate the adult organism. Um, there's another process called cell specialization, and that is after division. Uh, and you get two daughter cells. In many cases, those daughter cells have different properties, and I'll show you an example of that. Maybe make that a little clearer in a minute. There's also interactions between cells, and again, so some cells are signaling other cells to carry out certain functions or to go through certain changes. And then finally, cell movement. And so there are a lot of rearrangements of cells during development, and the head is particularly a place where there's a lot of rearrangements, and it's made it a little more difficult to study the head because of that. But we find ways to get around that. So let's look at this cell specialization idea. And so up at the top in this figure, you see a parent cell on the left, and it produces some kind of determinant. And then that determinant, shown by the red dots here, um, can be sequestered into one area in the parent cell, so that when it divides, one of the daughters gets all these determinants and the other daughter cell doesn't. And so then you see you clearly have an asymmetric division here where the two daughter cells are not the same anymore. Now at the bottom it shows another way to get differentiation of two daughter cells, and that is if one of the daughter cells comes in contact with uh, signals from outside that confer a different mode on that daughter cell versus the other one that doesn't get the signals. But let's go back to the first example, or first idea here, the asymmetric division, and look at an example of that. And this is from the small worm C. elegans. This is a nematode that when it's totally mature, just over 100 cells. So it's a very um, simple or multicellular organism, and every worm develops exactly the same way, unless there's something wrong with it or some kind of mutation. And that sequence of development has been mapped out specifically so that we know where the lineage of every cell in the adult worm. But this is an example very right after fertilization, where in the top left we see the sperm coming in, and then the nuclei or the DNA is stained blue, so that in the second panel we see the cell has divided into two cells, and each one of them has a nuclei, and then we see further divisions as we move to the right. Now in the bottom panel, these bright green dots are what are called polar granules, or P granules, and these are involved in developing the germline for the next generation. So if you don't have these P granules, and they're not just in C. elegans, but in other organisms as well, if you don't have them, the organism will be sterile. And so these are only going to be found in the germline. And so you can see that as, well, even on the left in the initial single cell, the P granules are migrating to the posterior end of the cell, so that when it divides into two in the second panel, all the P granules are in the right-hand cell. And then as they divide further, you see they get sequestered into a smaller and smaller space. And finally, in the last panel, they're still in just a single cell. Now, of course, that cell is going to divide, and we'll get multiple cells that make up the germline. But this is an example of specialization and how a single cell, without any influences from the outside, can divide into two very different daughter cells that carry out different functions. Now, another process that's going on, as we mentioned a minute ago, is signaling. And this is a schematic showing how this works. If you have a set of cells shown in black at the top, they can emit or secrete signals that go out and affect the surrounding cells. Shown in the bottom in blue are those cells that were affected by the signal. And generally this will set up a gradient so that the cells closer to the signal receive a higher signal than those farther away. So they may be affected in a different way than those that are seeing a lower level of the signal. And so the presence of these signals is not only important in a certain time and place, the level of the signal or the amount of the signal is important as well. So if you increase the signal, and that is the signal molecules that are secreted from the signaling cells, you're going to spread that signal out over more cells and affect more cells. If you decrease the signal, you'll have the opposite effect. So you can see when you get a highly complex organism like humans, subtle changes can have some effect. Can make people taller or shorter or whatever, depending on the level of expression of some of these signaling molecules. Now, a third process that's going on is, again, cell movement. And this is an example of that. And now we've switched to another model system, frog, Xenopus. This is an embryo shown on the left, a schematic showing the embryo at the 32 stage. And at that stage, individual cells were injected with different colored dyes. And these are pretty big cells at this point, because the cells, or the embryo in the frog is fairly big, and so you can inject these big cells reasonably easily. So you can see then we have a blue cell, a pink cell, and a green cell. And then if we come back 10 hours later, now there's about 50,000 cells. And you can see how intermingled some of these cells are because they still contain the dye. And so you can see uh, some of them look like a multicolor, and that's because there are cells on top of each other that are different colors that give that sort of orange color at the very tip um, of the embryo here. But again, you can see um, the amount of rearrangements going on even very early in development, whereas later in development, there's probably even more of that sort of thing going on. So we have cells dividing, so proliferation, we have the cells moving around, we have cells signaling to each other to carry out different functions, and we have cells dividing asymmetrically to give two daughter cells that have different functions. Now there's one more concept I want to talk about, and this is the idea of cell determination or fate. And you can see, at some point during development, cells are going to be fated for a particular tissue. Okay, so before that, they can be developed into many different types of tissues depending on their surroundings and other signals. But at a certain point, they've received the signal to be a certain type of tissue, and so there's no going back under usual conditions. And this diagram shows an example of that. So on the left we have a developing embryo shown as a gray oval here, and so no visible sign of differentiation. And at some point, then we start to see visible differences, and this is shown in below that, whereas the one end of the embryo is green, the other end is red. Now, if we go back and take some tissue out of that embryo before we see any kind of overt morphological differences, and we move it, in the case in the middle here, from one side of the embryo to the other. If it's early enough in development, it won't make a difference in the development of the organism because those cells have not yet been fated or determined. So they will develop as any other cell in the vicinity, in their new location. Now, if on the other hand those cells have already been determined, then if you put them into another location in the embryo, they're going to develop as if they were in their original location, and so that's shown by the coloration in the bottom here, where cells were taken out of what was going to be the red area and put over in the green area, but they don't develop as green cells, they now develop as red cells. Before they were taken out of the donor cell, which again had no visible sign of differentiation, they were already fated to be red cells. So even if we move them over to the green area, they still are going to develop as a red cell. And I'll show you an example of that. So an example of cell determination is shown in this diagram in another model system, in this case the chick. And in this example, tissue from the leg bud, and that's just a sac of cells that are again, showing no morphological development into a leg, but these are cells that are going to be developing into a leg. If we take some of those cells out and we put them into the wing bud, again, a group of cells that are going to develop into the wing, what we find is that the adult chick has developed foot-like or claw-like features at the end of the wing, where the wing should be. So what this is showing us is that those cells that were taken out of the leg bud, even though they showed no sign of differentiating into different leg structures, were already fated to be leg cells, and so moving them, in this case, to the wing bud did not change that. So those cells developed into foot-like structures rather than wing-like structures. So again, this is a case where the cells were moved after the cells were determined or fated to be a particular tissue. So they were no longer responsive to signals from the resulting or the surrounding tissues. Now, another interesting thing, again, I've mentioned that there's a lot of similarity in these developmentally genes from one organism to another, and this is an example of that. Looking at the gene called engrailed, and this was a gene we'll talk about in a few minutes that was first identified in the fruit fly Drosophila and is involved in early development as well as later on in development. But in this example, I should mention that there's an engrailed gene also found in the mouse as well as humans. And in this example, we see on the left, in the left panel, a normally developing mouse, and the mouse brain and the cerebellum is identified here. If we have a mutation in engrailed in the mouse, that cerebellum fails to develop properly. You can see the structure looks quite a bit different. It just fails to develop fully. However, if we insert the Drosophila or the fruit fly engrailed gene into the mouse genome, now the cerebellum develops normally. So you can substitute the mouse engrailed gene with the Drosophila equivalent and get normal development. So these genes are doing very similar things in both these organisms, and in this case, even though Drosophila don't have a cerebellum in their brains. So I want to talk quite a bit now about Drosophila, because this has really been a good model system for studying development, partly because well, it's easy to manipulate in the lab, and the turnaround time is just a couple of weeks for each generation, and also because it has a lot of morphological features that can be identified and often in a dissecting microscope. Again, the worm C. elegans has been a very important model system because it is so simple, but there aren't very many morphological features. Also, there's very little of cell-cell interaction going on in C. elegans. As I mentioned, each organism or each individual worm follows a strict cell lineage, so there's no variation from one worm to the next, whereas that's not the case in Drosophila. There's a lot of cell-cell interactions going on, so it's more similar to a mammal or a human, where there's also a lot of interactions going on. Now, another powerful tool in Drosophila are shown here, what are called polytene chromosomes. And in many of the embryonic and larval tissues in Drosophila, you get giant cells. And in order to accommodate those big cells, there may be many copies of the genome. And so most of the larval cells are polyploid. Mainly those cells that are going to go on to be in the adult fly are diploid, but the rest are polyploid. And having these large cells eliminates the need for a very complex vascular system in a larva, which is a transient form of the organism. So in some cells, particularly in the salivary glands, the polyploid chromosomes line up next to each other based on homology and form what's shown here, these polytene chromosomes. So this is in just a regular microscope looking at 100x, and you can see the entire Drosophila genome here. And it has a very distinct banding pattern, and each one of these bands has been identified. And unless there have been some rearrangements or deletions or insertions in the fly genome, every fly has this identical banding pattern. And like I said, each one of these bands has been identified. And people that work on these polytene chromosomes, if you showed them a little segment of the polytene chromosome, they could tell you where it is in the genome based on the banding pattern. So this allowed people working on Drosophila to identify the location of many of these genes using genetic tools, and looking at deletions and insertions for one thing, and asking when the gene was there or not, depending on what had been deleted. The other interesting use of the polytene chromosomes is to study gene expression, because as you can see here, as we move from the left panel to the right panel, we see that parts of the polytene have been expanded, and those are called puffs, and those are due to expression of the genes in that region. So you can study gene expression by looking at when certain regions of the genome are puffed, and when they go back down. And these were some of the first really gene expression studies, because before there were any molecular tools, you could study expression, and people studied hormone induced expression, because you could pull these salivary glands out of the embryo and study them in a culture dish. So they looked at hormone stimulation of expression, and also the changes in expression due to what's called heat shock, or heating in this case, the cells up to a temperature that it's not happy with, and it expresses these proteins that are there to deal with stressful situations. Anyway, that's a bit of a digression. I want to get back to development now, and again looking at the fruit fly, Drosophila, and at the bottom of this diagram is shown a larva, and you can see again it has a bunch of hairs on the bottom of it that can be used to identify different segments. And Drosophila, like most complex organisms, is a segmented organism, and we can divide it into these different segments. And so shown at the top is the embryo, showing the head segments shown in blue, the thoracic segments shown in red, and then the abdominal segments shown in green. And so there are 14 segments in the fly. And it turns out that actually the developmental unit for the fly is halfway between the segment, what we identify morphologically as a segment. And so those have been termed parasegments. So parasegments are offset by segments by a half. The segments being defined by morphology, whereas the parasegments defined by function or by genetic means. Okay, so the basic idea now is to set up this axis from the anterior to the posterior end of the embryo. There's another axis going from dorsal to ventral. I'm not going to talk about that today. It uses another system that's also interesting, but I think this makes the point. And so we're going to look at a series of genes that define smaller and smaller regions across the anterior-posterior axis of the organism. And then finally, once those regions have been defined, then a set of genes that will determine what develops in each one of those regions. And this is a list of them, starting at the top with what are called polarity genes. And these establish gradients along the length of the embryo. Now this is the one area that's likely different in Drosophila compared to mammals. And the reason for that is in Drosophila, after fertilization, the nuclei divide several times before they form cells. And that allows these determinants to flow freely in and out of the nucleus. So you can set up these gradients in the developing embryo before there are cell membranes between each of the nuclei. And so the nuclei are able to see these determinants, in this case the polarity genes. And once those gradients are established, then another series of genes come on in different region, and these are called the gap genes that define large regions in the embryo. And those then, along with the polarity genes, are going to turn on a set of genes that start to identify these segments. And these are called pair rule genes, and they're expressed in alternating segments. So there are 14 segments in the developing fly, and so each one of these pair rule genes will be expressed in 7 regions along the fly. And there are 8 pair rule genes that are each offset by 1 cell. Once those are established, then another group of genes, the segment polarity genes, are expressed in half of each segment, and so are defining even smaller region. And this is where actually the engrail gene is expressed. Originally, it's a segment polarity gene, but it's also needed later on in development in the fly as well as in humans and other mammals. And then finally, the homeotic genes, again, that determine specialization of each of these segments. So once we go through the first 4 groups of genes and define actually each half of a segment, then the homeotic genes are expressed to tell each one of those regions what to become. And so this next diagram depicts that, starting at the top again with the polarity genes and showing an example of one called bicoid, which sets up gradient from the anterior end to the posterior end. Then the gap genes are next and define large regions within the embryo, and two of them are stained for here, one called Krupel and one called Hunchback. And so you can see one is green and one is orange, and where they overlap in expression, you see a yellow area. A lot of the names of these genes are going to be derived, are going to have German names, and that's because a lot of these genes were first identified in a lab in Germany, and so they were given German names. Next, we go to the pair-ruled genes, now that are expressed in one segment wide bands, so 7 bands across the embryo. And these, in this example, we see 2 complementary pair-ruled genes. So we see 7 blue stripes and 7 orange stripes. At this point in Drosophila development, each one of these segments is 4 cells wide. So again, there are 8 pair-ruled genes that are offset from each other by 1 cell. So there's 1 gene for each of the 8 cells that make up 2 segments. In this case, though, we have the 2 genes stained for Eve, or it stands for even skip, and the other one often called FUTZ. FTZ stands for Fushi Terazu. It's a Japanese name, not clear where it came from. I worked in the same lab with a woman many years ago, Barbara Wakamoto, who had done her graduate work studying the genetics of Fushi Terazu, and everybody assumed, since the name is Japanese and Barbara's last name is Japanese, that she had named the gene. But Barbara grew up on a farm in Arizona and does not speak a word of Japanese and had no idea what Fushi Terazu meant. Okay, so next then we go from the pair-ruled genes to the segment polarity genes. And again, these are expressed in half of each segment, so we get 14 stripes instead of the 7 stripes we see with the pair-ruled genes. And these have, well, maybe we'll come back to that in a minute. And finally, then the homeotic genes that are expressed throughout the embryo that define each region and tell it what to do. Now, I've gone through this in pretty detail, but again, we want to just think about the concept here and the idea of having groups of genes that define smaller and smaller regions in the embryo until finally those regions can be told what to develop into. Okay, so that's the bottom line here, and I don't think we need to really focus on the specific genes here. I mean, we could memorize all these genes if we wanted to, but I don't think that's really necessary. Again, showing a little more detail, the expression patterns of the 2 complementary pair-ruled genes, the even skipped and Fushi Terazu, at 2 different times in development. And again, these both make up 7 stripes along the anterior-posterior axis of the developing embryo. Now, again, this is the example of a segment polarity gene, showing 14 stripes at 3 different times in development. Now, unlike the earlier genes that are expressed early in development, the segment polarity genes are also expressed in the adult, as you can see here engrailed as when you use a stain that's things for engrailed, it shows the 14 stripes along the adult fly. The other difference between the segment polarity genes and all the other genes, or the upstream genes, all the upstream genes, all three groups of them, are all DNA binding proteins that affect the expression of different genes, mainly downstream genes. Whereas the segment polarity is a mixture of DNA binding proteins, of which engrailed is one, and signaling genes, either those genes that are going to be found on the cell surface of one cell are a receptor for a signal, or those gene products that are going to bind to those receptors and signal cells. So again, during the first few rounds of these developmental genes, we're talking about DNA binding proteins, but when we get down to the segment polarity genes, then we're talking about both DNA binding proteins as well as cells that signal other cells or receive signals from other cells. Now here's some examples of what happens if you have a mutation in these genes, and basically for both the GAP and the parallel gene, deletion of the gene will cause a lack of development in the region where that gene is expressed. So you see with the parallel gene, if we delete the Krupel gene and its expression area shown in green here, then we see that the embryo develops without that area. So that area where Krupel is normally expressed fails to develop. Now, of course, this embryo is not going to live very long, but it does develop into a larva, a non-viable larva. The parallel genes, again, expressed in every other segment, and so a mutation in parallel gene will cause the development of only half the segments, so you get only 7 segments instead of the normal 14. Mutations in the segment polarity genes are a little more complicated. What you get is deletion of the anterior part of the segment and duplication of the posterior part in the mirror image of the regular posterior part of the segment. So a little more complicated. But basically you can see that if you don't express these genes in the right place, those areas of the developing organism are not going to develop. Now I want to talk a little bit about how these genes affect gene expression, how we develop these complex patterns of expression, starting here with the polarity genes. And this shows 2 ways to set up a morphogen gradient in an embryo, like a Drosophila embryo. And one is to express a gene at the very end, in this example shown at the anterior end, and let it diffuse towards the posterior end, forming a gradient. The other approach is to express that gene throughout the embryo, but have an inhibitor that's expressed at the posterior end that inhibits it, or that forms a gradient from the posterior end, inhibiting the morphogen over the gradient of the inhibitor. The result is a gradient of the inducer in both cases. Now, again, there are 2 gradient systems set up in the developing fly. One that for the anterior-posterior axis, and that's shown at the top of this figure, where a bicoid on the right is expressed at at the anterior end and forms a gradient towards the posterior end. And then another gene called nanos is expressed at the posterior end and forms a gradient in the other direction. There's also a gene expressed at both ends called torso that affects this as well. And then there's, in addition to that, another gradient set up in the dorsal-ventral axis of the embryo, and this is through a gene called TOL. You may have heard of TOL or maybe TOL-like receptors that are important in the innate immune system. And in fact, it's the same gene, although again in humans there are an array of TOL-like genes. So TOL in the Drosophila is used to set up this early dorsal-ventral gradient. So it's expressed on the ventral part of the surface of the embryo and forms a gradient. But it's also used later on in the Drosophila immune system. And uses the same pathway of expression. So in this case, TOL is used for two very different things in the fly, and again is found in humans as well as other mammals in multiple copies that carry out the same function in the immune system. But today we're more interested in development, or its role in development. But again, we're going to go back to the anterior-posterior axis and not really talk about the dorsal-ventral development anymore, except to say if you decrease the level of TOL receptors, if you lower the expression of TOL, you will dorsalize the embryo, so the tissues will be more skewed towards the dorsal side. If you increase TOL, you will ventralize the embryo. And again, so this is an example of not only expressing the gene at the right time and place, but the amount of expression can change the development of the organism as well. Okay, back to the anterior-posterior axis. And again, how do we set up these complex patterns of expression? And so this is an example of expression of one of the pair rule genes. Remember, the pair rule genes are expressed in 7 stripes along the axis, the anterior-posterior axis of the embryo, and that's shown in the bottom right-hand figure here, the 7 stripes of the EVE or even skip gene. And if we look at the gene itself, what we see is that there are different modules, regulatory modules, that are promoting expression in the different, in the 7 different stripes. So we can take one of those modules, in this case, the module for the stripe 2, and it's shown on the bottom left here, put it upstream of a reporter gene. This is just simply a gene that when it's expressed turns the cells blue. And so when we put that back in the fly, we see that it expresses, or you get one blue stripe where the second even skip stripe is. Okay, so this region upstream of the coding region for EVE is directing the expression of the even skip gene in the stripe 2 of the developing embryo. Now, there are four genes that are involved in regulating that module, the STRIPE2 module in the Drosophila embryo, and these are the four genes. The bicoid gene, remember, that's a polarity gene that sets up a gradient from the anterior to posterior axis, and then three gap genes, giant, hunchback, and Krupel. And this is their pattern of expression. And so if we look at in the next figure, the expression of these four genes along the anterior-posterior axis of the embryo, showing particularly Eve STRIPE2, what we see is that in the region of the STRIPE2, hunchback and bicoid are both expressed. And so those are positive regulators of Eve, whereas giant and Krupel are expressed outside of the stripe and are negative regulators of Krupel. So of Eve. So giant and Krupel turn off Eve, hunchback and bicoid turn on Eve. And so by setting up these patterns of upstream genes across this region of the embryo, we can get expression of Eve in this STRIPE2. Now if we look at the schematic of the actual regulatory region for STRIPE2, what we see are a number of binding sites for these four different proteins. And again, Krupel and giant are repressors. They turn off the gene. Bicoid and hunchback are activators. They turn on the gene. And you can see there is some overlap here of the repressors with the activators. So depending on the concentration of both of these, you're going to either get binding of the activator bicoid or the repressor Krupel. And so that's how you can regulate the expression of, in this case, Eve over this narrow stripe. And so each one of these stripes has its own regulatory module that responds to these upstream regulatory proteins in a different way. So we looked at that last example in pretty detail, but the details aren't important, you know, the names of the genes or whatever, but the concept that you have these cascade of genes that are narrowing down the regions on the embryo to smaller and smaller areas is what's important, and also that these upstream genes can be expressed at different levels across the embryo to produce a pattern that allows the expression of these genes that are expressed in a pretty complex pattern, like the example we just saw of 7 stripes across the embryo. Now, once these areas are defined or outlined, now we need some genes to tell them what to do, and that's the homeotic genes shown here. The list of genes, or the schematic of actually the chromosome on the right here. And in Drosophila, the homeotic genes are found in 2 clusters, the antennapedia complex and the bithorax complex. We'll see in a minute that in humans they're all in the same complex. But the other interesting thing is they're expressed at the same in the same order that they appear on the chromosome. So you can see the expression patterns here on the left, the segments or parasegments shown at the top, and where these different homeotic genes are expressed along the anterior posterior axis. And so if we look at a schematic of the embryo, we see again that they're expressed at different regions in the embryo. And if you look closely, you'll see that many of or some of the segments have more than one homeotic gene expressed there, and that's what's going to give it its ultimate form. So they do combine to identify different regions on the embryo. So I've shown you this picture before of a fly with the legs growing out of his head instead of antenna, and this is a homeotic mutation where the antennapedia gene, a gene that's involved in or is expressed in the thorax of the fly and involved in delineating tissues in the thorax, is expressed in the head as well. And so what's happened is a chromosomal rearrangement that brought the coding region for the antennapedia gene next to a control region that turns genes on in the head. So you get expression in the wrong place. Of course, if you knocked out the antennapedia gene, that would be lethal. But this is a dominant mutation that causes a homeotic change. In other words, a change in the structures present in a certain particular region of this case, the fly. We can also get a four-winged fly. Now, the normal Drosophila is a two-winged animal, where there's on the wings are always on the second thoracic segment, the second of three. On the third segment are a set of vestigial organs called halteres. But by manipulating the genes in the bithorax complex, and it's a fairly subtle and complicated manipulation, you can get a fly with four wings. So it basically has two second or T2 thoracic segments instead of one. Both this fly and the Antennapedia flies are viable in the lab. In other words, you can mate them and propagate them, but I don't think they would last very long in the wild. Okay, so we've looked at the Drosophila homeotic gene cluster, and let's compare that now to the human homeotic genes. And what we see here is the Drosophila clusters. Remember, they are in two different clusters in Drosophila. At the top of the diagram, at the four lines at the bottom are the human clusters, because there are four copies of the homeotic gene complex. Each cluster is together, so they're not broken apart like in Drosophila. But you can see the redundancy here in the humans compared to the Drosophila. And again, this is another example where the genes involved in development are very similar across a wide array of organisms, all the way from flies on up to humans. But as a general rule, we often see more copies in the more complex organisms like humans, and in this case there's four copies of the homeotic genes. And there's a lot of redundancy built into that, but there's also some slight differences in the homologous genes from one copy to the next, so that if you knock one of the genes out, so if one of them gets mutated, there are some subtle differences, some subtle changes, but it's not catastrophic, not like knocking out one of the Drosophila homeotic genes. Now, between the two clusters, what is considered the ancestral complex that all these different organisms were derived from. And again, if we look at the expression pattern across the embryos, or in this case the adult fly, we see the same pattern in both human embryos and Drosophila. And again, this They are expressed in the same order on the developing embryo that they are found on the chromosomes in both flies and humans as well as other organisms. Now I just want to show you one more example of a developmental gene. This is called the KIT gene. And again, just to emphasize the fact that these genes are found across organisms and have very similar functions. And a mutation in the KIT gene, as shown here, causes a similar phenotype in humans as well as mice.